Hey everybody, welcome to supplementary video 18.5 to 18.6. So this is going to be a really short video, just 3 slides, I think, talking about hemostasis like we have in 18.5, and then getting a little bit into transfusions and blood typing in 18.6. So hemostasis is the process of closing or stopping blood loss. You can think of it as a blood vessel has been broken, and that's a bad thing because now you are losing blood. If you lose too much blood, you get low blood pressure, which can cause all sorts of different issues from there. And so the body needs to be able to respond to this and stop the blood loss really, really quickly. So failure to seal, when you lose a bunch of blood, that is called a hemorrhage. So there's 3 steps that take place in hemostasis. You're going to start with vascular spasm. So that's going to be the blood vessel, which is made of smooth muscle, is going to contract. Um, uh, the blood vessels have 2 different directions of muscle tissue that they have. One of them is circular. So that circular one is going to contract, and that's going to close the blood vessel, or at least constrict the blood vessel. And the other is longitudinal, so it runs along the length of the blood vessel. And so when that contracts, it's going to pull it away from the site of injury, or it's going to withdraw it from those tissues. Actually, that longitudinal muscle withdrawing that blood vessel can actually make it difficult for surgeons if they're trying to find blood vessels to be able to reattach or to close them, pinch them off, and seal them off, and things like that. But obviously, our bodies are not considering the possibility of a surgeon taking care of that blood vessel, and so that withdrawal reflex does occur. After the vascular spasm, you're going to have a platelet plug start to form. So platelets or thrombocytes are going to encounter the rupture. They're going to react with some of the chemicals that are released when that vessel ruptures, pain chemicals and things like that are flowing through, and that's going to cause the platelet to change shape a little bit, and it's going to become very spiked and very sticky, which means it's going to have a better ability to grab onto other platelets flowing by and release some other chemicals that are going to help with vasoconstriction, or the closing of that blood vessel. Once you have that platelet plug form, you'll then start to see coagulation, which is where you're going to see all sorts of clotting factors, and we won't go into them. There's 12 steps and 2 different pathways of clotting and coagulation. But you're going to get a blood clot that starts to form from the fibrin, starting to tangle some platelets and starting to tangle erythrocytes in that coagulated area. Vitamin K, I will say, is very important for that cascade pathway. Which is why when a baby is born, pretty much right after, they're going to give the baby a shot of vitamin K. Too much vitamin K is not going to hurt the baby, but if they have any sort of bleeding from the trauma that is birth, vitamin K is going to help provide, or that extra vitamin K is going to make sure that they have enough to be able to stop any sort of bleeding that that baby may have. And then a long time after, you're going to get fibrinolysis, which is where that fibrin is going to start pulling the edges of the rupture together. Think of it like when you have 2 ends, or you have a shoelace in both hands, you pull it tight, how that pulls your shoe, the sides of your shoe closer together. That's kind of what's happening in fibrinolysis when it pulls the edges of that clot together. And eventually that clot is going to degrade, and the tissue around it hopefully is going to heal, depending on what type of tissue it is. Blood types, we talk a little bit about these on the high school portion of the class. Remember that an antigen is going to be some sort of identifying marker. Usually we talk about surface antigens. So these are going to be markers on the outside or on the membrane of the cells. Antibodies are going to be things that are identifying and calling out foreign particles. For your blood and for ABO and RH, or especially ABO, these antibodies are going to be Y-shaped. They're going to bind to foreign particles when they run into them and make them start to clump together. It's called agglutinate. There's more than 50 different antigens on your red blood cells, but ABO and RH are going to be the most important. When we are testing for somebody's blood type, we'll take a sample of blood and we'll separate it into 3 wells. So we'll have, this is all blood from the same person, just put into 3 different wells here. In one well, we're going to put anti-A antibodies. So that means these are antibodies that are considered type A antigens foreign. The second well, we'll put anti-B antibodies in. So these are going to be ones that consider type B to be foreign. And then anti-D antibodies, which is actually what's going to be testing for that Rh factor. If the antigen is present, that means the anti-antibodies did encounter that antigen. You're going to start to see that agglutination, that clumping to occur. So we see here that when we put anti-A in, we do have a reaction. So that tells us that this is type A blood. No reaction with type B, so we don't have an AB type. And when we put anti-D in to test for that Rh factor, we do have a reaction. So we know that this is positive because it does have the Rh factor. Okay, so that's just a quick addition to the high school portion of this side, talking about hemostasis, the sealing of a blood vessel to make sure we don't lose too much blood, as well as taking a look at blood